ellipses correspond to the 95% confidence intervals of the groups. And so what we can see here is that there is a clear difference, a clear separation between dogs that, in terms of the microbiome compositions of dogs that are not aggressive from those that are aggressive. This was a small pilot study, but it provided some foundational knowledge for the field to move forward and really begin to understand whether or not the microbiome is linked to aggression. This, of course, is just a statistical association and does not provide any evidence that the microbiome is causing aggression. With all that being said, the mounting amount of information is now pointing to the microbiome's relationship with behavior in neurophysiology. Just again to summarize, it seems growingly clear that microbes in the central nervous system can communicate through the gut-brain axis. Well, another interesting point to be aware of is that one's microbiome is not static. Our microbiome is dynamic, it's mutable, it can change. We, through different types of decisions or lifestyles or environments, behaviors, can make decisions that affect the composition of our gut microbiome, and sometimes in very dramatic ways. Imagine a real hyperbolic example is in the case of taking an antibiotic. If you consume antibiotics, you're going to eradicate certain members of your microbiome that are sensitive to the antibiotic, and you're going to enrich for microorganisms that are not sensitive to the antibiotic. And that is going to have at least a temporary, if not long-term effect on the types of microbes that live in your guts. Well, the foods that you eat, other pharmaceuticals, and even environmental pollutants can all have relatively similar types, maybe not necessarily quite as severe, but can have similar types of effects on the composition of your microbiome. And so your gut microbiome is sensitive to a variety of environmental stimuli. And our lab and many others have spent the better part of the past 10 years really trying to understand what types of environmental exposures can perturb the composition of the microbiome. And it does appear to be a relatively sensitive component of our bodies. Whether or not it is resilient, meaning it could return back to its original state upon cessation of the stimuli, is something that is still kind of being sussed out. But it does look like the different types of decisions that we make have an impact on the types of organisms that live in our guts. Collectively, this leads to, I think, to a very interesting and provocative question that we need to disentangle. And it's this: could the microbiome mediate how our environments impact our behavior development? So for example, if we know that there are environmental stimuli that affect behavior, but we don't understand how, and we know that these environmental stimuli can impact the microbiome, and if we know that the microbiome can coordinate with the central nervous system, is it possible that at least some of the effects of the environmental stimulus on behavior are occurring by way of the microbiome? This is interesting to consider, because if that's the case, then we may be able to diagnose certain types of mental health disorders through analysis of the microbiome, with the gut microbiome, which is relatively straightforward, inexpensive, and non-invasive to do. And alternatively, it means that if the microbiome does appear to be the central link between environmental stimuli and behavioral dysregulation, then perhaps we can develop microbiome therapeutics, which are sometimes also referred to as psychobiotics, that allow us to improve or treat different types of mental health disorders by manipulating the microbiome or mimicking its biological effects. Now, these observations are not just limited to behavior, and there is evidence to indicate that environmental stimuli and environmental exposures may affect the gut microbiome to impact behavior and cognition. For example, as I've already alluded to, environmental exposures can affect the gut microbiome and behavior in correlated ways, at least in many animal models. We see this routinely in zebrafish and in mouse when we challenge these organisms to different types of neurotoxicants, for example, and nutritional variation. These associations include measures of cognition such as learning, memory, and motor skills. And what you see on the right-hand slide, again, is one of these principal coordinate analyses that I mentioned earlier. But here we worked with Jacob Raver's group at OHSU, Oregon Health Sciences University. And we looked at mice that were given a neurotoxicant. And these mice, this neurotoxicant, for reasons that are not well understood, elicits different types of cognitive impairments that are consistent with neurodegeneration, including Alzheimer's disease and Parkinson's disorder. And what we find is that the presence of the neurotoxin, or the consumption of the neurotoxin in the diet, also affects composition of the gut microbiome. Now, again, this doesn't show cause and effect, but it does suggest that there is a consistent relationship here that is worth disentangling. And we also know that, as I've mentioned, microbiome manipulations support, at least in some instances, a causal role for the microbiome in behavior. And so there does appear to be a mounting array of evidence in support of this question, this hypothesis that I posited. And so what I want to do with the remainder of the talk is share with you some recent work where we've dug in to this question, specifically around the topic of neurodevelopment and the potential role that the microbiome may play in mediating the effects of the environment on neurodevelopment in children. And so just to provide a little bit of background in the case, this isn't a well known problem in your circle. You may be aware that childhood mental health disorders are increasing, at least in the United States. This does appear to be something that is occurring with increased incidence in developing nations or developed nations around the world. And this is a problem because we, first of all, don't understand the basis for this rise. And second, a child's behavior affects their ability to socially integrate. It affects their opportunities for success, and it predicts their lifelong tendencies for behavioral dysregulation. Certainly that's not universal, but children that experience behavioral dysregulation have an elevated risk for behavioral dysregulation over lifespan. And so these observations have motivated an interest in trying to define the processes that underlie perturbed behavioral development. But this has proven to be very challenging simply because the problem is so complicated. But one area that has received a lot of recent attention is the role that the early life environment plays in shaping behavior development. And this has been motivated by a variety of observations. First and foremost is that neurophysiology can develop extensively throughout childhood. And this creates a prolonged window for environmental stimuli to affect the development of neurophysiology and behavior. And indeed, researchers have been able to demonstrate that exogenous factors appear to drive behavioral development. And this can include things like nutrition and antibiotics and the psychosocial environment. We're going to talk a little bit more about the psychosocial environment in a moment. Yet in almost all cases, we don't understand how these factors get under the skin. How do they elicit their effects on behavior development? And this remains a pretty big open question. And there are probably a variety of different ways through which they do so. Well, I want to zoom in on the psychosocial environment because this is an area that has received extensive attention and for which there is a very strong amount of evidence that demonstrates that the psychosocial environment that a child experiences does have a fairly strong effect on the behavioral development. And just so that we're all on the same page, the psychosocial environment is basically the set of social factors that a child experiences that affect or that interact with thought and behavior. And this can include things like family nurturing and support, social capital, how well the child integrates into their social networks, the trust that the community has in them, and vice versa. It can be things like the child's social inclusion or exclusion. And this oftentimes can go hand in hand with things like socioeconomic factors, like poverty, deprivation, so on and so forth. And it also includes things like community capacities, resources, and resilience, to provide support structures for children. And again, this is something that is also connected to socioeconomic status. And there's now an extensive amount of evidence on the table that it demonstrates that socioeconomic and caregiving factors in particular, these are critical components of the psychosocial environment that a child experiences that drive normative behavior development. We don't know how. It's not clear. And there are probably a variety of different underlying mechanisms through which these exogenous factors impact behavior regulation in children. But we hypothesize that the gut microbiome may play a role here. And we think that this is a particularly interesting and exciting hypothesis to consider, because studies in animal models indicate that the gut-brain axis can impact neurodevelopment. And so we know, for example, that the assembly of the microbiome, like neurophysiology, is not something that happens instantaneously. It happens throughout childhood and adolescence. The gut experiences ecological succession, much like a forest does over its, the span of its ecosystem. When children emerge into the world, they do so with a sterile gut. And it's the inoculation of exogenous microorganisms that then colonize the microbiome, assemble into a microbial community, and then diversify as that child experiences the world and interacts with different parts of the world. Their immune system and their physiology develops. The microbiome diversifies and takes on a more complex form. This diversification of the microbiome early in life appears to be pretty coordinated in many instances with the development of neurophysiology. And so this suggests that there may be this linkage between the development of our gut microbiomes, the development of our brains by way of the gut-brain axis. It's pretty speculative at this point in time, but there is some data supporting this line of work. And so what we sought to determine if there's credence to this hypothesis, and if early life changes in the microbiome in particular could yield lifelong impacts, or excuse me, the implication of exploring this hypothesis is that it could indicate that early life changes on the microbiome could yield lifelong impacts on not just the microbiome, but on gut-brain function. So one concern that we have as a research group is that if there is extensive crosstalk between the development of the brain and the development of the microbiome, and since the microbiome is sensitive to exogenous factors like pollutants, then it's possible that the exogenous environments that we experience can disrupt our microbiomes in ways that elicit altered neurophysiological development early in life that have long-term lifespan complications on behavior, mental health, cognition, etc. This is a hypothesis that we intend to explore. And to get there, though, we have to answer some foundational questions. And the first is, how does the gut microbiome link to behavior in children? This is not something that's very well studied in humans. Well, we also need to determine if the gut microbiome modulates a child's behavior, how a child's environment impacts their behavior. Sort of a central tenet to this idea that we have. Does the microbiome appear to be a mediator of environmental effects on behavior early in lifespan? And what are the potential mechanisms through which the microbiome may execute its role as a mediator of environmental impacts on behavior? So to address this question, of these sets of questions, we've elected to apply our bread and butter techniques as a research group, which is a systems biology approach. And this is where we conduct multi-omic investigations, where we apply high throughput DNA sequencing approaches with computational and bioinformatic procedures and data analytic investigations to resolve connections between different aspects of a host's environment, their genotype perhaps, their microbiome, and how they all relate to the host phenotype. Now, the one downside to the systems biology approach is it doesn't show cause and effect. Our goal isn't to necessarily say this causes that, but our goal is to understand the linkages, the statistical connections between these complex processes so that we can then drill in and develop hypotheses that we can drill in and test with mechanistic follow-up experiments. And today I'm going to tell you about a recently conducted systems biology approach that we executed to address some of the prior questions. Now, one additional innovation that I want to point you to here is with respect to methodology. Most microbiome studies apply something called 16S sequencing. And if you've ever picked up a microbiome investigation, you've probably seen that phrase leveraged. And the way that it works is you take stool from a patient, which contains a variety of different types of microbial cells. You extract all the DNA from that stool, and then use polymerase chain reaction to target a specific gene that's present in basically all the different microorganisms, or in this case, bacteria, and then you sequence the amplicons that are produced by PCR. And these DNA sequences that come from a distinct gene that is different in each of the different organisms that comprise the microbiome, these DNA sequences then serve as a digital fingerprint of who is present. That's all that it tells you. It tells you what organisms are present. Well, remember, we're interested in trying to understand what these organisms might be doing. What are their biological functions and how do these functions relate to environmental exposures, and how do they relate to behavior? And so we applied something called a metagenomic approach. All right, and the way that this works is, again, we start with a stool sample that's got a variety of different microorganisms inside of it. We extract DNA from all those different organisms, and then we apply something called shotgun DNA sequencing, which is the same approach that is used to sequence whole genome sequences, except we don't just have one genome. We have hundreds and hundreds of genomes. And so what we end up with are a variety of very short DNA sequencing reads that are randomly sampled from along the length of the genome for the myriad genomes present in the community. And what we then have to do is bioinformatically characterize, well, which of these little DNA sequences might come from genes, protein coding sequences. Now, some of these some of these sequences are going to come from genes that are taxonomically diagnostic, like the 16S gene. And we can analyze those short DNA sequences and make inferences about who is present. Some of them, however, are going to come from protein coding sequences, genes that encode a protein that does something. Maybe we know what that protein is capable of doing. And as a result, we can make inferences about what these organisms are doing metabolically, or likely doing metabolically inside of the microbiome. So this is the approach that we used in a recent study that sought to determine how socioeconomic risk and caregiving behavior, collectively, which we refer to as a psychosocial environment, relates to the gut microbiome and its functional capacity, and how the gut microbiome's functional capacity relates to behavioral dysregulation in a variety of children. This was a relatively small study. It's not an easy study to do because it's hard to get access to this patient population. We had about 40 children who were preschool aged. And what we did is we got a microbiome stool sample from these children, and we had parents fill out a variety of surveys that were then screened by our experts, by our colleagues who are experts in psychology, child psychology. And these surveys allowed us to quantify various categories of socioeconomic risk, various measures of behavioral dysregulation, various measures of caregiver behavior. And we were also able to collect information on demographics and gut history, which allowed us to control for things like antibiotics and diet to ensure that the associations that we're measuring are not simply due to those variables. We then applied metagenomic sequencing to all of these stool samples. And what we ended up finding is now summarized in this part of the presentation. So the first observation we made is that the gut microbiome's functional capacity does associate with the psychosocial environment. And this is the first time that this result has been documented in the literature. Here, what we see again in one of these, this is basically similar ordination, the principal coordinates analysis, except this is a conditional correspondence analysis, but it basically shows the same information. Again, every point is an individual child, and the distance between them is effectively how similar the microbiomes are. In this case, though, we're annotating points in two different ways. The first is the internal black-white gradient indicates the amount of dysfunction in the relationship between a caregiver and a given child. The outer ring corresponds to a socioeconomic indicator of the number of turmoil life events that a child has experienced according to the surveys. And what we end up finding is that there is a relationship between the dysfunctionality of the relation, there's an association between the relationship of the dysfunctionality between a parent and a child. And there's also a relationship between the microbiome's composition and the number of turmoil events that a child experiences. And these two different terms statistically interact as well. And that interaction also relates to the composition of the gut microbiome. So in other words, this indicator of socioeconomic risk, life event turmoil, the number of turmoil events a child experiences, and caregiving behavior, the type of bond between the parent and the child, both link to the types of metabolic pathways that are encoded in the gut microbiome. We also find that the functional capacity of the gut microbiome links to behavior in these children. Here, we show this for a variety of behavioral measures made in this study, but here I show you the results specifically for impulsivity, impulse control. And what we end up finding is, again, every point is a child, and this color gradient corresponds to their impulsivity score. But children that are more impulsive carry different types of biological functions encoded in their gut microbiomes than children that are less impulsive, okay? What we also find is that there is a relationship, an entanglement between inhibitory control of behavior, parent-child dysfunction, and the functional capacity of the gut microbiome. Again, the internal points correspond to the relationship between the parent and the child. The external rings correspond to the inhibitory control. And what we can see here is that these terms statistically interact to explain the variation in the functional capacity of the gut microbiome, suggesting that these terms are all intertwined with one another. So what we then sought to do is not necessarily to look at things at this 10,000-foot view, but to drill in and look at specific functional pathways that are intertwined with the psychosocial environment and with behavior. And what we end up finding using our regression approach that allows us to account for various statistical properties of the gut microbiome is that there are biological functions, metabolic pathways encoded in the genomes of the microbes that comprise the gut microbiome. And that for some of these pathways, their relative abundance in the microbiome correlates with measures of behavior, for example, such as fear. Okay, so we conducted this type of pairwise regression approach for a variety of measures of behavioral dysregulation. And for all the different types of functional modules we were able to detect in the gut microbiome. And we end up finding there's a rich array of interactions between different functional modules and different measures of behavior dysregulation. We did the same thing for functional modules and socioeconomic risk, and also find that there's a diverse array of interactions. So to summarize our results here in this analysis, we find there are about 690 microbial protein families. You can think of these as components of metabolic pathways in the gut microbiome that link to 7 different psychosocial environmental variables and 13 different behavioral dysregulation symptoms. Of interest, we note that these microbial protein families tend to be enriched for different types of high-level functions. This includes pro-inflammatory functions. We see things like type 2, type 3, and type 6 secretion systems that are known to induce immune activation in the hosts, being associated with behavior dysregulation. And we also see things like heme transport modules, which are oftentimes also linked to immune activation in inflammatory bowel disease patients. We also know that there is an association between monoamine precursor metabolism and these different types of variables. Tryptophan biosynthesis and metabolism is a consistent player in a variety of different behavioral dysregulations and socioeconomic risk factors, as are things like glutamate, leucine, and glutamine biosynthesis pathways, and these are known monoamine precursors for a variety of neurotransmitters. And so collectively, what these results seem to imply is that the biological functions of the microbiome are intimately intertwined with various measures of behavioral dysregulation and various measures of socioeconomic risk in ways that are somewhat consistent with our expectations. And this, it's very tempting to take these results and, now that we have been able to link various aspects of the psychosocial environment, the gut microbiome, and behavioral dysregulation, and intuit that what these results indicate is that that socioeconomic risk and caregiving behavior affect the microbiome in ways that affect behavioral dysregulation. That's a tantalizing and provocative hypothesis, but we can't disentangle it because of the analytical approaches that we're using from an alternative hypothesis, which is that the psychosocial environment is independently affecting behavioral dysregulation, and that because the central nerve, because the gut-brain axis is reciprocally interacting with the gut microbiome, that behavioral dysregulation is then affecting the gut microbiome. We can't disentangle these hypotheses yet, but our follow-up work seeks to do so by using experimental models in a reductionistic setting that allows us to determine the causal pathways through which these intertwined relationships are acting. It's also worth noting that it's possible that socioeconomic risk and caregiving behavior independently affect the microbiome and behavioral dysregulation, an alternative hypothesis that we need to explore. Regardless, the study clarifies the potential to utilize the gut microbiome as a clinical resource. In particular, the functions that we identify here may prove to be fruitful leads for determining the microbiome's impact on child behavior, and could be potentially useful in a clinical setting to predict a child's behavioral or developmental trajectory. But this is a small study size, and we need to really expand it to be sure that these are robust results. We also need to think about longitudinal measuring to determine if the changes in the microbiome temporally predict subsequent changes in behavioral dysregulation. It also suggests that we may be able to one day develop possible therapeutics through microbiome manipulation that help restore problem behaviors into neurotypical states. But there's a long road before we're there. This work also, however, indicates that context matters, that our ability to develop a lot of these clinical resources, like diagnostics and potentially therapeutics, has to consider the environmental context, specifically the psychosocial environmental context of a child, because children in different psychosocial environments carry different microbiomes, and the child's relationship between the microbiome and behavior is fundamentally different as a result of that psychosocial environment. And so it also suggests that clinical resource development around the microbiome for a neurological perspective is going to be complicated to develop because of these environmental effects. Regardless, we ultimately hope that this study and others like it will potentiate the development of additional research that allows us to really drill in and understand the mechanisms through which the assembly of the gut microbiome are contributing to the development of brain functioning throughout childhood and adolescence, and how different types of environmental factors come in and interfere with that typical development. So we're running short on time, and I just want to finish by sort of summarizing a few key points that if you walk away with nothing else, I hope you walk away from today's presentation with these ideas. And the first is that many microbes are actually good. They contribute to our homeostasis, our neurotypical behavior and brain functioning. And this is, the work in microbiome research is upending this old legacy of perspective that the microbes that we encounter in the natural environment are germs that need to be nuked from orbit. Instead, what we're learning is that the microbes that reside on our bodies play an important and intimate role in shaping our physiology and behavior. And so we need to think about how we cultivate beneficial microbiomes when we consider various aspects of the clinical experiences of our patients. The microbiome is also a relatively mutable health component. And so we also have to think about how our environment and our lifestyles may disrupt the gut microbiome in particular to drive mental health and neurological disorders, and whether or not our understanding of mental health and neurological disorders can be advanced and improved by deepening our knowledge of the microbiome. Ultimately, the future of mental health care might include evaluation and management of a patient's gut microbiome. However, my personal view is that we are in the really early days of this field, and extensive research is required before we can arrive at that destination. That being said, I think there's some very exciting leads that are coming out in the literature, and this is a field to keep an eye on and potentially contribute to, because it seems like there may be opportunities to really think about transforming mental health care through consideration of the microbiome, given the data that has been generated thus far. I want to thank you for your attention. Again, I'm sorry about the hiccup. I mean, that's the Zoom era for you, right? But I really appreciate the invitation to come out and to speak to you all today and to interact with you. I'm grateful for the people in my lab that have helped to propel this work, as well as our collaborators at the University of Oregon. The work that I mentioned in terms of the childhood behavior development was a very excellent partnership with Phil Fisher, who's a behavioral psychologist of children, and a very talented PhD student at the time, Jessica Flannery, who collaborated with a postdoc in my lab, Keaton Stagaman. I've also benefited enormously from a collaboration with Jacob Raber and colleagues at OHSU around the role the microbiome may play in neurodegeneration. And of course, we're grateful to our various funding agencies for propelling our work forward. So thank you very much. I'll leave it at that. And if there are time for questions, I'd be happy to field them.
Thank you very much, Dr. Sharpton. This is a wonderful presentation. Actually, that was an error from our side. We apologize for that one. Apparently, Google told us it was 10 a.m. in Oregon, not 9. So we will.
What can you do? Yeah, I'm glad we were able to connect, and I'm glad it worked out. Thank you very much.
Thank you very much for being so quick. So we will take some questions now. One first question from Dr. Howard Cherkov. How do you deal with the fact that diet changes in psychiatric disorders?
Yeah, that's a great question. I mean, so we do our best to model the dietary effects that we observe in our data. And so basically the way that it boils down, for those of you who are sort of statistical junkies, the way that it basically works is what we do is we first model the effect of diet, and then we fit the residuals, the remaining variation between the microbiome. So we produce a model of diet, how diet explains variation in the microbiome, and then we fit the residuals with the remaining covariates that we're interested in studying. The challenge, of course, comes into play when we run into situations where diet is perfectly confounding with psychiatric measures. And here, this ends up being very challenging. In such instances, we actually do not observe the psychiatric effects because the diet variation slurps up all the explanation of variance for that variable, and we miss the effect. So it's quite possible that we're under discovering psychiatric effects that exist because we're allowing the nutritional information to be the first order variable that explains that variation. That being said, that's the conservative approach. That's the approach we prefer to apply because we know that diet has a huge effect on the composition of the microbiome, and so it seems appropriate to allow that framework to explain the effects. What you're seeing in this study as a result are effects that do not appear to be attributable to diet. I should also note that in this particular study that I've reported here today, all of the children are actually subclinical. All right, so they're not. This is an important point of distinction that I meant to articulate and failed to do so. These children are subclinical. So we see a spectrum of scores, but none of the scores are actually reaching clinically diagnosed dysregulation. And so what we're seeing then, so in the case that there are really sharp distinctions around nutritional variation and those disorders. In our data, we're not actually reaching the point in our analysis of the data, at least, where we see complete confounding between behavioral state and nutrition, at least in this population of very small children. But it is a problem that needs to be dealt with in the field. And unfortunately, from a statistical perspective, our options are very limited. It's a great question.
Thank you very much for your answer. One comment from Olivia Tartastet, fascinating research, amazing approach as a bioinformatician interested in mental health, circadian cycles and gut microbiome. This is truly inspiring, thank you. And one question from Edeltraud Kröger, is it time yet to do trials on probiotics and anxiety disorders?
So first, thank you for the prior comment, and then thank you for this question. Well, I think the question about is it time to do trials on probiotics and anxiety disorders is a great one. There are, from what I recall, there are some studies that are currently exploring this. I think the data on probiotic administration in humans is so far pretty thin. I think the work so far suggests that there's probably merit to the approach. And in preclinical studies, particularly in mouse models, we do see, as I referenced in the presentation, we do see effects of probiotic supplementation on phenotype, both of all different sorts, but including behavior in animal models. And so it suggests that there's a future for probiotic implementation in humans as well. However, a lot of the probiotic clinical studies that have been conducted thus far have been pretty inconsistent, and it's been difficult for probiotics to really zero in on clinical effects that appear to be robust. And some of this probably has to do with the contextual efficacy of probiotic implementation. When we conduct a preclinical trial in a mouse model, the mice are in a very fixed environment. It's controlled environment, it's controlled diet. And the probiotic, in many instances appears to respond well. But when we get into humans where there's a lot more variation, this tends to be much harder to control. And it's not always clear that the mechanisms of the probiotics that we're implementing in human studies are necessarily the right ones. Should we deliver freeze-dried cells? Should we deliver functional foods that contain probiotic taxa alongside prebiotics? Should they be probiotic cocktails? The long story short here is that I think we're kind of at a point in the field where we need to work out the right technologies for probiotic implementation alongside searching for the specific sets of taxa that should be subject to probiotic supplementation, maybe before we get to the point where we're really thinking about extensive clinical trials in probiotics. That being said, my personal view is that there's a sufficient amount of data on the table to justify moving into clinical trials around probiotics. I think, though, that these questions of implementation are going to remain barriers to entry in terms of peer review of those ideas. And they probably need to be really well considered before moving forward with them from a federal funding perspective, in my opinion.
Thank you very much. Yes, so one more question from Amanda Rishk. Do you take into account whether children are born via a natural birth or via a C-section?
Yeah, good question. So there is a bit of a debate about whether or not the route of birthing affects the microbiome. And so some of you may be familiar with some of the work that has argued that if you're born through the vaginal canal or through a C-section, you tend to carry different gut microbes. And certainly early in life, that appears to be the case. There is some debate about the legacy of that signature on the microbiome. The skeptics of the claim have argued that once the child gets onto breast milk and then solid foods, that a lot of those effects become pretty minuscule. But there has been some subsequent work that has gone on to show that children as of 10 years of age still maintain modest, but effects nonetheless. Our study admittedly did not account for variation in birthing strategy. And so it's possible that if there's a relationship between natural versus C-section birth and these psychosocial parameters or behavioral tendencies, that could be a confounding study factor that we need to better account for in our investigations. But my personal view is that the effects that are attributable to birthing strategy are pretty modest compared to the effects that we're seeing in this investigation. That being said, it's a fair chink in the armor of the analysis.
Yeah, definitely. Okay, thank you very much. One more question from Asana Shokri. There are emerging studies suggesting a link between the ALS and gut microbiome. Do you think this will have a great impact in the field of ALS research?
That's a really good question. I mean, ALS research is really complicated, and it's been really challenging to resolve the underlying factors that are driving the disease. From the data that I'm seeing in the ALS field, I think the answer is yes. I'm not totally sure that the microbiome is the trigger. I think the right way to be thinking about all of these diseases and disorders is basically health is a multivariate equation. genetics, diet, the toxicants you're exposed to, all contribute to health state. And what we're learning now is the microbiome does too. And in some instances, it also interacts with these other terms in the model to affect health state. And I think the question is, what is the loading on the microbiome's turn? And from the data that I'm seeing in the ALS research, it suggests that the loading is pretty meaningful, at least in terms of disease severity, possibly because the microbiome really, particularly the gut microbiome, really does play an important role in modulating the immune system. And so I think there's probably an important relationship here. And I know that there are a rapidly growing number of researchers that are looking into this particular question. I think there's a lot of merit to it. We've been working with the Raber lab to think about the microbiome's role in Parkinson's disorder and Alzheimer's disease. And we are seeing, again, very intense or intimate entanglement between the microbiome and disease symptoms, or rather phenotypes that are consistent with disease symptoms in mouse models. And these include humanized mouse models where we've taken disease alleles from humans, put them into the mice, and we see that the mice that carry the disease alleles carry different microbes, and that those microbes then go on to explain the specific conformation of those microbiomes within the diseased allele population, explains variation in the behavioral phenotypes that those diseased mice then display. So we think that there is something with neurodegenerative diseases and disorders in general in the microbiome. I suspect ALS will not be an exception there, particularly given the intimate immune relationship between the disease and the microbiome.
Thank you very much. And one more question. So apparently, it seems that the number of gut microbiomes in our gut is 7 times more than the number of cells in our body. So if this information is correct, apparently we have something like 7 humans in our gut that influence us every day, our decisions and emotions. Do you think, or as per your knowledge, are there any distinct group of microbes that concentrated on certain functions, like some microbes influence only anxiety, other microbes influence only depression, something like that, or it's more like everybody works on everything?
Yeah, that's a great question. So the first is that we've seen this number sort of conjecture that they're 7 to 1, 10 to 1. That number goes back to a 1972 study where there was some pretty extensive extrapolation, and recent reassessment actually puts the number of microbes in the gut on a little over 1 to 1 to human cells. Okay. Which, of course, there's everybody's favorite joke. That means that you become more human when you go to the bathroom. But another way of thinking about it is not so much that there are more bacterial or microbial cells in the gut than human cells, but there are far more microbial genes in the gut metagenome than the human genome by a factor of 300. And so these microorganisms, maybe they don't outnumber us cellularly. Maybe they're about equivalent with us cellularly. They outnumber us metabolically. They're capable of doing a very diverse array of things. What we are hoping to ultimately understand is who is doing what, and whether or not there are functional guilds that interface with our physiology along the lines of what you're conjecturing. We don't know enough yet to know the answer to that question. What it appears to be is that there are kind of usual suspects that come up in these investigations as being associated with neurotypical individuals or being associated with behaviorally dysregulated individuals. And what we think is going on is that there are biological functions that these gut microbiomes execute that are kind of distributed, because bacteria in particular don't just inherit gene functions, they acquire it horizontally from other organisms, which is how antibiotic resistance has become such a big problem. And so we think that there are different bacteria that have acquired different functions that are they're beneficial for our physiology in a variety of different ways, and that affects the central nervous system in sort of in a generic way, and that there are then other organisms that elicit more specific effects on central nervous system functioning, and we seek to determine who they are. Since we are seeing these common players associated with health and neurotypical behavior, this does give credence to the idea that we'll be able to develop probiotic therapeutics at some point. Those are obviously the ones that we want to prioritize as potential probiotics. But it isn't totally clear at this point in time. There just hasn't been enough investigation in enough populations, I think, to be able, across disease states, to be able to address your question with any more precision than this. So it's a great question. I just don't think we're there as a field yet.
Thank you very much. Okay, well, 300 is still a big number.
Yeah.
for the proteomics.
Right. Yeah.
So,
right.
Dr. Sharpton, I apologize once again for this with the time.
Oh,
Thank you once again for this marvelous, excellent, thought-engaging presentation. Now we know that we're not alone. And thank you once again for accepting our invitation. And it was a great pleasure to listen, to understand your research, and to see the perspectives of microbiome analysis.
Well, thank you very much for having me. It was I had a lot of fun. Thanks for the questions and the engaging discussion. And I really appreciate the opportunity.
Thank you very much. Thank you very much, everyone. So this would conclude our conference for today. Please refer to Dr. Sharpton's lab webpage, where you can find more information and directions for a potential collaboration. And thank you very much, and have a good day.
Thank you. Take care.